Welcome everyone to this exciting 30-minute video lecture on the tools of genetic engineering. Today we will explore the ingenious devices and techniques that have revolutionized our ability to manipulate the genetic code of living organisms. Let's dive in and unlock the secrets of these powerful tools. In the last lecture we have covered Introduction to Genetic Engineering, what are GMOs or genetically modified organisms, brief step involved in their creation, examples of various GMOs which have been created till date, their applications, risks involved and ethical concerns. Before we delve into the tools, let's quickly review the building blocks of genetic engineering, and it is the DNA, which is the blueprint of life. Humans and all other living things have DNA which contains hereditary information and that are passed from one generation to another. The information in our DNA gives our cell instructions for producing proteins. Proteins drive important body functions like digesting food, building cells and moving our muscles. It is basically the proteins which gives messages to the cell for various functions whatever we do. Your DNA is the most unique and identifying factor about you. It helps determine what color your eyes are, how tall you are, how dwarf you will be, how likely you are to have certain health problems. Even so, over 99% of DNA sequence are same among all individuals. It is the remaining 1% that explains much of what makes you you. It is this percent only which define individuality of a person. That's why in spite of being the same DNA, we differ from one another. DNA is arranged like two intertwined ropes in a double stranded way, and this is known as double helical structure. We can refer the figure for example. Here we can see each strand of DNA is made up of 4 types of bases or molecules which are basically referred as nitrogenous or nucleotide bases. These bases are attached to one another in a particular strand with the help of sugar phosphate backbone, or more specifically I can say they are attached with the help of a phosphodiester bond, and these 4 bases are adenine, guanine, cytosine and thymine. Adenine is abbreviated as A, the thymine is abbreviated as T, guanine as G and cytosine as C. These base pairs, they pair in a specific way across the 2 strands of DNA, or we can say these 2 strands they are complementary to one another. Like there is a thumb rule, adenine will always pair with thymine with a double hydrogen bond, and guanine will always pair with cytosine with a triple hydrogen bond. This is the basic thumb rule for maintaining the double stranded DNA or complementarity of the DNA double helix. DNA manipulation is a broad term. Herein we are going to study the tools that are required for DNA manipulation. This basically encompasses various techniques that are used to modify DNA sequences in a controlled manner. These techniques are commonly employed in fields such as genetic engineering, biotechnology and molecular biology. Some of the key methods used for DNA manipulation include PCR, that is polymerase chain reaction, DNA cloning, DNA sequencing, site directed mutagenesis, CRISPR-Cas9 gene editing, RNA interference or RNAi and synthetic biology. We will discuss all these methods in detail in subsequent lectures individually. Now the thing is how the purified DNA is manipulated. We will study in the next lecture how we will purify the DNA or extract the DNA. For now on, we will say how the manipulation of the purified DNA is done. To produce recombinant DNA molecules, the vector and the DNA to be cloned needs to be modified. It has to be shortened, lengthened or copied into new DNA molecules. It is modified by the addition or removal of specific chemical groups. It is basically a chemical reaction we can say. Many of the DNA manipulation techniques make use of purified enzymes, which within the cell participate in essential processes such as DNA replication, transcription, repair, recombination, breakdown of unwanted foreign DNA. There are various ranges of DNA manipulative enzymes, and they are DNA modifying enzymes, which includes nucleases. Again, they can be categorized into endonucleases and exonucleases. Second modifying enzyme are DNA polymerases, and the third one is enzymes that acts on termini. The next range of DNA manipulative enzymes are restriction endonucleases, or in short they are also known as molecular scissors, and the last one is again very important, that is ligases, which are also termed as molecular stitchers. Then coming to the nucleases, how they function we will discuss here. Nucleases, from the name itself, they targets the nucleotide bases, and ases, all the enzymes, they are always suffixed with a word that is known as ases. Basically, these ases are capable of digesting a particular molecule. So, when we say nucleases, they are capable of digesting nucleotide bases, or in detail I can say they degrade the DNA molecules by breaking the phosphodiester bond that links one nucleotide to the next in a DNA strand. Exonucleases remove nucleotide one at a time from the end of a DNA molecule. For example, we can see here in the figure, this is an exonuclease. How it functions? From the name itself, it will remove the enzymes from the ends. So, we can see here initially we have a double stranded DNA, and these double stranded DNA, already I told, they are made up of nucleotides. In a single strand, these nucleotides are joined with the help of a phosphodiester bond, and these two double stranded, they are linked with one another with the help of a hydrogen bond. Okay. So, when the nucleases or specifically exonucleases are applied on this template DNA, what is happening? From the ends, the red ones indicates the nucleotides from the termini are being degraded, they are being removed from both the DNA strands, one at a time from both the DNA strands. Then endonucleases, they will break the internal phosphodiester bond within a DNA molecule. This is the major difference between the exo and endonuclease, and from the name itself it is clear. The examples of endonucleases are DNase 1 or deoxyribonucleases 1, that cuts both the single and the double stranded molecule. I will show in figure how it can act on both the single and the double stranded DNA molecule. The end result of prolonged DNase 1 action is a mixture of very short oligonucleotide. Here is an example in two parts where we can see S1 nucleases. In the first figure, it is acting on a single stranded DNA molecule. Since it is an endonuclease, it will remove the nucleotides from within the DNA strand. Then coming to the second half of the figure, we can see it was a double stranded DNA, and when the endonucleases was applied or S1 nuclease was applied, a nick is created. Why? Because at a time it can cleave only a nucleotide from a single strand, and once the nick is created, then the second strand is exposed for the nucleotide bases to be degraded by S1 nucleases. So, one by one both the strand nucleotides are removed, and we can say there is the separation of nucleotides between the two strands with the help of S1 nucleases. Then here is an example of DNase 1. It again acts on both the single stranded and the double stranded DNA, but the major difference is it can act on both the strands at the same time, not like that of S1 nuclease, where it is cleaving the nucleotide from one strand and further it is cleaving the nucleotide from the second strand. Here it is cleaving the nucleotides from both the strands simultaneously. Then coming to the next classes of DNA manipulative enzymes, they are basically known as polymerases. From the name itself, they will help in polymerization. How? These are the enzymes that synthesizes a new strand of DNA which is complementary to an existing DNA or RNA template. Most polymerases can function only if the template possesses a double stranded DNA, and that acts as primer for initiation of polymerization. So, before going into the basic reaction of these polymerases, I would like to define what is a primer. We can see here in the figure, primer are again a short stretches of oligonucleotides which have a free 3' hydroxyl end. So, these primers, they attach to the template DNA or RNA strand and carries out the polymerization reaction. Here is an example how we can see. There was a template strand, the primer gets attached to the 3' end of the template strand, and both the strand of the DNA, we know that they are anti-parallel. That is, one strand runs from 3' to 5' direction, whereas the another one will run from 5' to 3' direction. Okay. So, here we can see that ATG was a primer sequence, and the red one we can see that the newly strands are being synthesized complementary to the template strand. How? I already told that G always pairs with C or T always pairs with A. So, based on the bases which are present on the template strand, the complementary strand gets synthesized by adding 1,1 nucleotide, and since it has a free 3' hydroxyl end, so it will create the phosphodiester bond between these two nucleotides. Polymerases, they are routinely used in genetic engineering. One basic landmark example of polymerase is DNA polymerase 1, which was isolated from E. coli or Escherichia coli. So, here we can see that in the figure we have a double stranded DNA, in between a nick was there. So, these DNA polymerases, they can also be helpful for filling the nick, but there is another landmark of these DNA polymerases 1, that is, in spite of filling the nick, it does not stops at G itself, but it will replace the existing nucleotides. Once it gets started, it will create on synthesizing the new nucleotides complementary to the template strand till the template strand nucleotides are completed. Then we have another major example of DNA polymerase, that is clenofragment. So, here what is the benefit of this? That is, DNA polymerase 1 has a dual activity, we have seen in the last figure, DNA polymerization as well as DNA degradation. Degradation how? Because of the endonucleases activity, it is replacing the existing nucleotides. But here this clenofragment, we can see that it lacks the nucleases activity. So, what it will do? It only has polymerase activity, that is, in total the green and the red one fragments which are present in the DNA polymerase, the green one indicates the nuclease activity, whereas red one indicates the polymerase activity. But in case of clenofragment, the green fragment is missing, that is, the nucleases activity is not present. So, what will happen? It will only specifically fill the nick, it will not replace the existing nucleotides, just was the case with your DNA polymerase 1. Taq polymerase is another very specific polymerase, or we can say these are the thermostable polymerase which can even act at higher temperature. So, this is an enzyme which is basically used in PCR reactions, because the PCR is operated at various higher temperatures. The different steps operates at different temperatures. So, we need a stable thermophilic enzyme. Taq polymerase was isolated from Thermus aquaticus. The enzyme is thermostable, that is, it is resistant to denaturation when the heat treatment is applied. Other polymerases commonly used are PFU, PVO, deep vent, TMA, and these are isolated from Pyrococcus furiosus, Pyrococcus voisi, Pyrococcus species and Thermatogota martina respectively. Reverse transcriptase is another enzyme which is used for DNA modification. It is involved in replication of several kinds of RNA viruses. It uses RNA as a template. It is essential to the technique called synthesis of complementary DNA, or in short we can say it as cDNA. Reverse transcriptase is involved in replication of several kinds of RNA viruses. They uses RNA as template. It is essential to the technique called complementary DNA or cDNA cloning. In this figure, we can say how the reverse transcriptase work. Here we have RNA as a template, we have a RNA dependent primer, and we can see this reverse transcriptase, they created complementary strand of the RNA, and this complementary strand is basically known as cDNA. Why it is known as cDNA? Because it is synthesized from mRNA and it is the other strand which is as complementary as the RNA. That is why only the coding regions are basically copied into the new strand of cDNA. Then the enzymes that acts on termini, they modify the DNA molecules by addition or removal of specific chemical groups. We have already studied one example of this, that was exonucleases. Here the second example of the enzymes which acts on termini are alkaline phosphatase. It was isolated from E. coli and calf intestinal tissue. This enzyme removes the phosphate group which is present at the 5' terminus of a DNA molecule. We can see initially at the 5' DNA strand, double stranded DNA, both the ends have phosphate group. When it was treated with alkaline phosphatase, what we can see, these phosphate groups have been removed. Instead of that, to maintain the stability, the hydroxyl groups have been added with the help of this particular enzyme. Then the next class of enzyme, this is basically used to prevent recirculation of the vector DNA digested with a specific restriction endonucleases. From the figure we can see that a nick was created, and when this particular enzyme was used, it was not recircularized, so that it can be again inserted with the gene of interest to produce a recombinant DNA molecule. Terminal deoxynucleotidal transferase from calf thymus tissue adds one or more deoxyribonucleotides onto the 3' terminus of a DNA molecule. Here we can see from the figure that at the 3' end, deoxyribonucleotides are basically added, and as a result the hanging ends are produced. It is mainly used to add homopolymer tails to cDNA molecules in order to clone them into a vector. One of the most fundamental tools in genetic engineering is restriction enzymes. Let us explore how these molecular scissors allow us to cut and manipulate DNA. Restriction enzymes are naturally occurring proteins that recognizes specific DNA sequences and cut the DNA at those sites. These cuts creates fragment with either sticky or blunt ends, which can then be used to insert foreign DNA. Restriction enzymes are indispensable in genetic engineering applications such as cloning genes, creating recombinant DNA and preparing the DNA fragments for analysis. Purified restriction endonucleases allow us to cut DNA molecules in a precise, reproducible manner that are required for gene cloning. This discovery received a Nobel Prize, and the discoverers were W. Arbour, H. Smith and D. Nathan in 1978. It was one of the key breakthroughs in the development of genetic engineering. They are synthesized by many, perhaps all the species of bacteria. Over 1200 different ones have now been characterized till date. Now, let us turn our attention to vectors. the vehicles that carry foreign DNA into the host organisms. Vectors are DNA molecules which are used to transport foreign DNA into host cells. Common vectors includes plasmids, viruses and artificial chromosomes. We will discuss each one of them in subsequent lectures, and they have specific advantages, and depending on their characteristics and needs, their applications are varied. Vectors play a crucial role in gene cloning, enabling scientists to copy and produce large quantities of a specific gene of interest. The vector molecules, here we can see that they are cut at specific site, and in the second half of the figure we can see the DNA molecule containing the gene to be cloned, and that is indicated here as a red fragment. So, for both of them the same restriction endonucleases has to be used to provide cut, and this cut DNA is again inserted into the nick of the vector molecule. Then the next class of enzymes are ligases. From the name itself, they will help in ligation, that is, they help in joining the DNA molecules together. Ligation is the final step in construction of a recombinant DNA molecule. See, there is a nicked vector molecule. When it is combined with a gene of interest, then we obtain a recombinant DNA molecule in presence of DNA ligase. Basically, this gene of interest is joined or ligated into the nick of the vector molecule to produce recombinant DNA or rDNA. The mode of action of ligases. Within the cell, the enzyme carries out the repair of discontinuities that may arise in one of the strands of a double stranded molecule. This lack of phosphodiester bond may rise by breakage of cell's DNA molecules or during DNA replication and recombination. Ligases used in genetic engineering is again purified from our model organism, that is E. coli, when infected with the phage bacterium T4. In the test tube, purified DNA ligases can join together individual DNA molecule or two ends of the same molecule. That is why they are also known as molecular stitchers. Blunt end ligation is not very efficient because ligases has to wait for chance association to bring the ends together. Blunt end ligation should be performed at high DNA concentration. Ligation is usually performed at 16 degrees Celsius. Here in the example we can see the two blunt ended DNA, they are joined with the help of the ligases. Ligation of complementary sticky ends is much more efficient because the base pairing between the two molecules is a relatively stable structure for the enzyme to work on. Here in this figure we can see we have a sticky or protruding or hanging end of a double stranded DNA. So, when we have to join or we have to break the discontinuities between these two strands, then the ligases will be used, and the end result we can see here in the figure, and the ligation will be much more efficient in comparison to the blunt ended ligation. Putting the sticky ends onto the blunt ended molecule. Linkers will be used. What are linkers? They are short pieces of double stranded DNA of known nucleotide sequences and are synthesized in vitro. We can see here in the figure the use of linkers. They have a protruding end. So, what they will do? They will create the polymer tail in the blunt end, and in this way the blunt end will be converted to the sticky end with the help of linkers, and the two ended DNA can be easily joined. Then the next class of short oligonucleotides are adapters. Again they are nucleotides with one sticky ends, and here they will help the ligation of the two different types of DNA strands. Other advanced tools that include gene synthesis. Gene synthesis is a technique that allows us to artificially create the DNA sequence, offering vast opportunities for designing custom genes and organisms. Then the second advanced tool is RNAi. It is a powerful tool that can selectively silence some specific genes, providing insights into the gene function and potential therapeutic applications. Now, let us have a quick recap of the tools that we have studied so far. Initially, we have a range of DNA modifying enzymes. The different DNA modifying enzymes that we have studied are nucleases. In these nucleases, we have studied exo and endonucleases. Then the second category was restriction enzymes. The third one was ligases. What are the specific functions of all these enzymes? Nucleases, they act on nucleotides. Then restriction enzymes, they again act on nucleotides, but they have a specific recognition sequences. And the third one is ligases, they are helpful in joining the two DNA strand, and to help the process of ligation we have adapters and linkers. Then there are certain other enzymes which we have studied, that is alkaline phosphatases and many more. These alkaline phosphatases, they add phosphate group at the ends. Apart from that, we have studied vectors, how the nick is created in a vector and the DNA fragment that needs to be cloned are incorporated into the vector molecule to produce recombinant DNA molecules. In conclusion, the tools of genetic engineering have transformed the various areas like biology, medicine and agriculture that offers incredible potential to address various challenges our world is facing, like all the challenges which we have discovered in the first lecture. How to create insect resistant plants, how to create pest resistant plants, herbicide tolerant species, as well as many applications in terms of animals to increase the yield, to increase the breed of the animals, or to enhance the milk production or egg production in animals, or even using these plants and animals for several medicinal aspects. As we continue to refine these techniques and push the boundaries of genetic engineering, it is very essential to use them responsibly, keeping all the ethical considerations in mind which we have discovered in lecture 1. Thank you for joining us on this enlightening journey through the tools of genetic engineering. I hope you feel inspired and eager to explore this captivating field further. Then, in the next upcoming lectures, we will study what are the steps involved in genetic engineering. Like I already told that these enzymes, they are isolated in the pure form. So, we will discuss in detail how these enzymes are isolated or how the purified enzymes can be obtained, and the other steps further for the creation of recombinant DNA till it is incorporated into host, or further expression of these proteins which these genes are carrying in large quantities in terms of bioreactor. Looking forward for the exciting journey, we will explore this field together. Thank you.